Okay, so we're going to spend basically the next hour going over the research, teaching, and teaching opportunities in our department and the facilities and capabilities that make our department special and allow our students and faculty to do their work. I have links to a couple of websites right here on the title slide. This is the website I've already given to you today that has all the material for prospective students, and this is just the general home site for our website, which surely you have all visited by now. Okay, so to start off, I just want to give everyone a feeling for what the priorities and philosophy of our department are with respect to how we go about graduate training and mentoring our students. And so the way we have structured things and what really backs our day-to-day operations is an emphasis on providing our students with the technical, the critical thinking, and the problem-solving skills that they need to succeed. And when I say they need to succeed, I just don't mean that they need to succeed and navigate our program and ultimately get a PhD, but what they really need to succeed professionally. Right? What do they need to get where they want to go, right? Because all graduate students ultimately have different priorities, have different aspirations, have different career goal in mind, and also have different personal goals in mind. And so we take that into account too, right? The way we try to do that is multi-pronged, right? The first part of our program that tries to take into account this personalized approach is through use of practical coursework that's flexible and that's independently tailored to enhance each student's foundation in the areas that are important to where they want to specialize. Okay, so we don't have just cookie cutter curricula where, for example, all organic students that come in need to take the exact same set of classes, right? All physical chemistry students that come in need to take the exact set of classes. Okay, we meet with our students once they have decided that yes, they do want to join our program. We have a conversation with every student independently to figure out, what are their foundations coming in? Where are their interests? Where do they ultimately want to go? What kinds of research are they interested in pursuing? To try to come up with a curriculum and set of coursework in their first semester and first year that makes the most sense based on those specifics. Okay. All our students have access to cutting edge research facilities, which is where they will spend the vast majority of their time with us as they pursue their PhD. We emphasize research and dissertation projects that push the boundaries of our field, okay? That aren't just staid, but that are groundbreaking in general, and have an emphasis on making real world impact, okay? Then on addressing either topical problems of importance or application based, and that incorporate collaboration across multiple disciplines. Right, across multiple subdisciplines within our fields of chemistry and biochemistry, and also other fields, right? With our sister departments like chemical engineering and material science and biology and biomedical engineering, and the like. We emphasize faculty mentorship that's focused on the development and maturation of each individual student, right? Our emphasis on this individualized or personalized approach doesn't end when we just talk about coursework and coursework is out of the way, right? Ultimately, when every student joins a research group, as every student works on a dissertation project, right? Their independent needs, their independent aspirations, they're still there. And so as we work with our students on their dissertation projects, as we mentor and train them toward their ultimate goal of obtaining a PhD, this is always in our mind, trying to give each individual student exactly what they need to get where they want to go, right? And finally, we try to provide all our students the opportunity to gain real world training and research experience with an eye toward long-term career development, okay? And we'll talk a little bit about some mechanisms that exist to do that toward the end of this presentation. All right. So just a few details on our campus and our university. So I really regret that we are not able to bring you personally on our campus because if you look at what our campus looks like online, it is absolutely a gorgeous place to be. The Princeton Review has dubbed UD as absolutely one of the most gorgeous campuses anyway. Where it's a beautiful place to be able to come and work and study every day. University has a long and distinguished history. Its roots go all the way back to 1743. So the roots of our university actually predate the birth of the United States. And UD is a major R1 university of moderate size, right? So there are about 1,200 faculty on our campus, roughly 24,000 undergraduate students, roughly 24,000 total students, close to 20,000 undergraduate, close to just under 5,000 graduate students. So we're a moderately sized campus. We're not as big as some places, like a University of Michigan or Ohio State, which have upwards of 60, 70,000 people on campus during the height of the semesters. But we're not a small place either, right? So there's that size, that gravity of a modern R1, but we're small enough where, in general, people don't fall through the cracks, and we get to have much more of a personalized approach in the way we deal with each other, and most importantly, deal with the students that we work with every day. Hopefully, at this point, 2021, we can all agree on who the most famous alumnus of the University of Delaware is. He's been in the news an awful lot in the last 6 months. It's this guy, our current president, Joe Biden. And so we're very proud of that fact. And I will say, I haven't seen President Biden on campus on the last year, but during my time at the University of Delaware, I'm in my 12th year now as a faculty member here. I have seen Biden on our campus at least half a dozen times. Right, this is a picture that was taken at commencement. Right, I've actually stood 12 feet away from him as he basically gave an overview of sort of strategic initiatives relating to energy development in the U.S. in the 21st century. Right. So Biden is a force has been on our campus a lot, and he may not be around as much, but I do expect to see him in the next 4 years. And maybe if you're not so much into politics and you're more into football, maybe you would say Joe Flacco is our most famous alumni, and that's fine too. We're awfully proud of him as well. Now, the fact that both Biden and Joe Flacco are alumni raises an interesting question, right? So UD is on a very select list of universities that have produced both a president of the United States and a Super Bowl MVP quarterback. Does anybody have an idea for how many universities are on this list? Take a guess. No? There's a list. It's only 4 schools. All right, so 46 presidents, 50-something Super Bowls, and there are only 4 schools that have produced one of each, and we're one of them. So we're awfully proud of that fact too. And it's a pretty distinguished list that you see there: us, Stanford, Navy, and Michigan. And if I want to raise the stakes a little bit, right, I can ask the question: well, how many schools have had a president, a Super Bowl MVP, and have had a faculty member that in the chemistry department that won a Nobel Prize in chemistry? And then the list is even more selective. It's just us and Stanford. So that's kind of fun. My point here is that UD might be a place that not everybody hears as much about, right? It doesn't have maybe the same obvious name recognition as a Stanford, for example. But UD is a place where great things have been happening for a really long time, and great things continue to happen. And there have been many, many, many people who have chosen to come to UD during their academic career and have gone on to do really fantastic things. And that's not a mistake. Okay, so where is Newark and where is the state of Delaware? In case you haven't had a chance to check us out, this is where we are. UD is basically located right where the circle is there. Newark, Delaware is almost exactly where the states of Pennsylvania, Maryland, and Delaware come together. So that tri-state border right there. In fact, from my home where I'm sitting right now, I can go on roughly a 5-mile run in a circle and hit all 3 of those states. Okay, so that's where we're located. We're right in the heart of the Mid-Atlantic region. Okay, so you can see that we're in close proximity to Philadelphia. Philadelphia, depending on how fast you drive, is roughly 45 to 50 minutes away. Baltimore is about an hour away, and Washington, D.C. and New York City are about 2 hours away. Right, so we're right in the heart of this Mid-Atlantic region, within easy driving distance of lots of major metropolitan centers, which is really nice if you just want to spend a day or a couple of days in a major urban center and take advantage of what those cities have to offer. What sort of distinguishes Newark and where the University of Delaware is that it's the I-95 basically runs right through here. So we're right on the I-95 corridor. Roughly 20% of the U.S. population lives on the I-95 corridor between Washington, D.C. And Baltimore, maybe as much as a quarter of the U.S. population by this point. But we're in basically this little protected area on that corridor that really has a little bit of a small town feel to it. Newark itself has a small college town feel that's right in the middle of this major area of population density. So it's a very easy place to live. It's a very relatively affordable place to live as compared to some of the urban locations around us. And our main street and the basic area around our campus really has that college town atmosphere, but we're not in the middle of a state and completely isolated and 3 or 4 hours away from a major urban center. So there's a little bit of everything where we are. As far as our city and the state of Delaware, there's really a little something for everyone in Delaware, from pristine beaches up on New Jersey, right? They call it the shore. In Delaware, it's the beaches, and Delaware sports some of the most beautiful beaches and summer vacation spots on the East Coast. Delaware has a really extensive state park system, which is phenomenal for anyone who enjoys spending time outdoors or doing outdoor activities regularly. So that includes hiking trails, running trails, biking, boating, camping, rock climbing. Basically all of that is within an easy 20 to 30-minute drive. Roughly a 10-minute walk from our core chemistry building is an entrance to White Clay Creek State Park, right near the our main gym, our athletic facilities, which offers tons of excellent hiking and running and biking trails, which I know many of our faculty and many of our students take advantage of on a weekly basis. About a 20-minute drive, if I back up a slide, right, I didn't mention Wilmington. Wilmington is a much smaller city than Philadelphia, has a roughly 150,000 people in it. But it's a wonderful place to be able to go for a day. It's got this really very exciting riverfront area, which is a great place to explore, to dine, to shop. A lot of fun there. Our state's minor league baseball stadium, the Blue Rocks, who are now a Washington Nationals affiliate, play there. And a little bit further to the south, about an hour away, if you're a NASCAR fan, we have Dover Downs, major NASCAR sites. So there's an awful lot to do in Delaware and the surrounding areas. All right, so I want to switch gears slightly now and start focusing on some specific aspects of our department. We'll start with just a few statistics. So on our campus at the University of Delaware, our Department of Chemistry and Biochemistry is amongst the earliest grantors of PhDs on our campus. Both we and chemical engineering awarded our first PhDs in 1948. So we've got a long legacy of graduate and doctoral education at the University of Delaware. Our department sports 30 or thereabouts research active faculty and research groups that students can join to pursue their dissertation projects. Our program size is roughly 180 or thereabouts doctoral students. It moves a little bit each year, but that's pretty much the steady state number. And students in our program going into next year will earn a graduate stipend of $28,500 for the 12-month year. Research programs in our department span across all of chemistry and biochemistry, as well as many tangentially related fields. And research in our department is linked to over $10 million in annual expenditures on external grants, which is a pretty fine level given the modest size of our department. As far as the physical space of our department goes, it's divided over 3 buildings that are right in the middle of the central part of our campus, the core of UD's campus. And the pictures of those buildings are shown here. Together, that gives us roughly 215,000 square feet for our research and teaching missions. The oldest of the buildings, built around 1930, was Brown Lab. Of course, its insides have been renovated many, many, many times since 1930. So no worries. If you come and join us and work in that building, you are not stepping back in time. Lamont, and so there are both teaching and research activities that take place in Brown Lab. This is Drake Hall, a much newer building. The 2 ground floors have a lot of teaching capacity, and the upper 2 floors are dedicated for research, and Lamont DuPont Lab, which is mainly for research activities. Having said that, things for our department and on our campus are far from stagnant. So the footprint of our department and our program continues to grow with new modern cutting edge buildings being built as we speak. So the first I'll mention is an expansion to Drake, one of those existing buildings that were on the last slide. So here was Drake. You can see the style and the architecture of that building here. Oops. So that's this area right there. And this entire new extension is currently being built. Ground has been broken. This area is currently fenced off. And this building is going up. And so that building is going to add additional capacity for our department's teaching and research missions. So there are going to be dedicated brand new teaching labs in that building that our faculty and students will get to use as they teach undergraduates. And there's also the plan for research space in that building as well. We're also super excited by this building, which has gone up on UD's Stark campus. So that's a modest 20-minute walk from say Brown Lab, one of those core buildings that I showed you on the last slide, or roughly a 4-minute ride on one of the UD shuttle buses. And so this building represents one of the new focus areas for science and research on UD's campus, which is in biopharmaceutical innovation. And so several faculty and several research groups in our department are going to be part of the inaugural team that opens up this building and commands space and actually gets to do their research. And we expect that the footprint that our students, our faculty, our department commands in this building will only grow in the years to come. All right. Speaking of our faculty, faculty in our department, they span all of chemistry and biochemistry, and they are all, I would say, well-recognized leaders in research and teaching and mentorship broadly across chemistry and also specifically within their subdisciplines. Here are some notable awards and notes that distinguish the faculty in our department. So, first and foremost is something that I've already alluded to, right? One of our former faculty, Dick Heck, was awarded the 2010 Nobel Prize, or a third of that 2010 Nobel Prize in chemistry for development of the Heck reaction, which we're awfully proud about. Will always be a point of pride for our department. Our department has multiple fellows of the American Academy for the Advancement of Science, the publishers of Science magazine. There are multiple members of our faculty that have won AP Sloan Research Fellowships. Multiple members of our faculty have won fellowships from the Dreyfus Foundation for their teaching and their research and mentorship activities. We have multiple Cottrell Scholars. Members of our faculty have won DuPont Young Investigator Awards. The majority of faculty that have come through our department in the last decade and a half to 20 years have won NSF Career Awards. Prior to that, members of our faculty have won Presidential Young Investigator Awards, and members of our faculty have also been awarded Pew Scholars for the Biomedical Sciences. So you should know that should you choose to join us at UD, you will have your pick, right? Multiple choices of faculty to work with in our department who are at the top of their fields, right? And that are well respected within their fields, which matters. Okay. Here's a snapshot of all the faculty, the names of all the faculty within our department. And on this slide, they're sort of divided within traditional subdisciplines: analytical, inorganic, biochem, physical, organic. Okay. What I want to emphasize to you is that you might find a representation or some slotting of faculty within these different groups on our website. We mainly organize ourselves in this way as a way to manage tasks, everyday tasks that we just need to do, and our teaching responsibilities in our department. Right? This view is not necessarily the way we think about research and training of our students day in and day out. Right? By contrast, the research in our program is really very highly interdisciplinary and collaborative. Right? And as evidence to that, if you watch this slide, if I animate, okay, the names that have changed to orange, okay, if you go to our website, those are names of faculty that each will be found under multiple traditional divisions, right? Because they might take care of teaching and sort of task management over in multiple areas, right? If I was to color code this and ask the question, how many of the faculty in your department have pursued research programs and mentor students whose dissertation projects are multidisciplinary and collaborative, it would be every faculty member in the department. And that's not just a sales pitch, and that's not a stretch to say. That's how we pursue science within chemistry and biochemistry at UD. And so in that light, we're going to spend a little bit of time now basically providing an overview of some of the core research areas that are important within our department. And so we're not going to present this as research from analytical groups and then research from biochemistry groups and the like. Okay, we're gonna talk about roughly 10 to 12 different modern topical areas of research, right, that cross-cut across our entire department. And you'll see the names of many faculty on every one of those slides, and on every slide you will see faculty across multiple of these traditional areas, right? Okay, and so with that, I'll toss it to Mary Watson, who will get us started off with transition medical catalysis. But before I do turn it over, let me also just say, just for the sake of time, we picked 10 to 12 topics to present to you. The science that you're going to see, that we're going to quickly go over the next dozen slides or so, is by no means, right, an exhaustive summary of all the science that goes on in our department. It's just some of the important topical areas that we're choosing to present to you today. So keep that in mind as well. Okay, Mary.
Thanks, Joel. So I'm really excited today to start off these topical areas by talking about transition medical catalysis. And the reason I'm excited about this is an area where my group works. And one of the best parts for my group of being at University of Delaware is that we're within a community of both organic and inorganic chemists that are focused in this area. And that makes my research program better because we're surrounded by people working in similar areas. So I wanted to point out a few of the things on this slide. So if you look at the top of this slide with this red powder and this beautiful red powder in this flask, this is an example from Professor Joe Fox's research group. And so if you've been following Joe's work, you know that his research program is really focused on the development of extremely fast bioorthogonal ligation methods that can be used even within humans now. But a huge part of that program is developing the reagents for people to use. And so what's shown here is actually a new reaction that his group developed using palladium catalysis to these Libeskind type cross couplings to enable access to these sorts of reagents. If you then flip to the bottom of the slide, you'll see another example of a cross coupling. This was a cross coupling catalyzed by nickel that was developed by Professor Don Watson's group. And this was just published, and it's this beautiful example where they do a dimerization event and not only make the carbon-carbon bond linking these two aromatic pieces, but also do it with nearly complete control over the three-dimensional array, giving nearly enantiopure products. And that's really important because the main application of these products is as chiral ligands for other transition metal catalyzed reactions. And so both of those give you a flavor of maybe the more organic side or organic application of transition metal catalysis within our department. But I'd be remiss not to mention really beautiful work in transition metal catalysis to solve the world's energy crisis that's being spearheaded by a number of groups, and probably most notably right now, Professor Joel Rosenthal's group. And so you can see those examples in the middle where he's using these nickel-based catalysts to convert carbon dioxide into reduced products that can then feed into fuel production or other resources to really combat this major societal problem. As Joel said, we had to be selective about what we include on the slide. So these are only a couple of examples of research in transition metal catalysis in the department. And I really encourage you to reach out and talk to each of the faculty on this slide if you're interested in this area.
Great, thank you, Mary. All right, so Eric Block will tell us about some materials related science in our department.
Thanks, Joel. Hi, everybody. Mary gave a very good background into the diverse types of chemistry we're studying. And broadly, as chemists, many of us are studying just materials as new ways to solve problems, from very fundamental studying materials, surface processes, to making and designing materials for applications ranging from energy to the environment to human health. So Joe's tetrazine chemistry that Mary brought up, an example of using this in a bioorthogonal approach. And the bottom right is using new materials to help image and diagnose catalyze reactions within the human body. Sort of the opposite end of this spectrum is Andrew Teplikoff's work, shown directly above this, is looking at catalytic processes on surfaces, which helps us understand and design brand new catalysts, largely focused on this small molecule activation problem. In this case, looking at the absorption of ammonia to a surface, which is a very important process, thinking about making fertilizer from nitrogen and hydrogen on a large scale. Definitely span from harder materials like solid surfaces. On the bottom left, showing one of Svillin-Bobev's clathrate complexes, which is very important materials for batteries, all the way to wearable electronics and biopolymers, which some of their features are highlighted here as well. If you could go to the next slide, please, Joel. And a real focus for all of us studying materials, whether they are on the softer side, biotype materials, through pure inorganic catalysts, is we are both developing new materials, using new methods to make these compounds, but also studying them in depth to really optimize and understand catalysis. For example, again, in the bottom left, Svillin-Bobev's work is highlighted, looking at the effects of structure on the function of magnetic properties of new classes of materials that he's making. Right to the right of this, Joel Rosenthal is looking at very interesting catalysis, not only on the molecular side, looking at reducing CO2, but looking on solid metal surfaces, in this case, mixtures of copper and tin, and how we can use ionic liquid additives to tune product distribution from things like CO to other important products. Emil is a new professor who's in his first year in our department. He's looking at making new nanomaterials, but importantly, developing a mechanistic understanding of how and why these materials form, which will help us tune catalytic properties, tune electronic properties. So we're really looking at trying to understand these materials from the bottom up to develop brand new processes. The last one here showing is a collaboration between Joel and myself, looking at making metal organic frameworks attached to electrode surfaces. This covalent attachment allows us to do a catalysis at an electrode surface, use it for sensing applications, large-scale separation processes. So we're really proud about this really cross-cutting and dynamic research that we're doing along the lines of materials chemistry.
All right, thanks, Eric. Catherine Grimes will tell us about how we use the tools of chemical biology to better understand disease.
Thanks, Joel, and thanks, Eric and Mary, for setting the stage for such dynamic research going on within the department. We're also really interested and have very rigorous program aimed at developing chemical tools to study important and relevant problems to human health. And I know that this is really important to many of you, as it was highlighted in many of your personal statements that you were part of your application to the University of Delaware. And I wanted to point out that this also drives the research selection of many of our labs within this department. And so two of our newest members of our faculty, Jody Hayden-Perella and Juan Perella, highlighted here, holding this Ebola virus, were recently in the New York Times talking about their work on modeling coronaviruses. And so I'm really interested in relevant tools to develop biophysical models and thinking about ways to combat something that's really relevant to our health right now. And then other labs, such as the Zhuang Lab and the Mudridge Group, are really interested in thinking about developing tools to study cancers. And so Jeff Mudridge's group, highlighted here on the top part of the slide in the middle, is really interested in thinking about how to tag specific RNAs. And then Zhihao Zhuang's group is really interested in thinking about this problem of ubiquination and how cells signal around certain proteins. And that theme follows through into Neil Zonlow's group, who's very, very interested in thinking about these small marks on proteins, such as nitrogenation, highlighted here, thinking about how these reactive nitrogen species play a role in Alzheimer's and Parkinson's disease. And we've heard many great things about this tetrazine reaction from Professor Fox's lab. And so highlighted here at the bottom is a new type of reaction that the Fox lab has developed. This is still a tetrazine reaction, but it gives them the ability to use light to activate this reaction in a spatial and temporal controlled manner. So you can really now see specific imaging within, here they're showing it in a mouse, but as it was mentioned, this can be done in a human body as well. And finally, on the bottom right-hand or left-hand side of the slide, my group is really interested in studying and developing bacterial probes to study the role of bacteria in cancer, inflammation, and neurobiological diseases. And so all of us are really truly focused on better understanding the mechanisms of diseases and then designing and then actually synthesizing the tools to do so. So it's really exciting applications into human health.
Awesome. Thanks, Catherine. Sharon Neal is going to tell us about science in the area of computational theoretical chemistry and data science as well.
Thanks a lot, Joel. So we have so we've highlighted on the slide several images to connect you with some of the computational, theoretical, and data-enabled chemistry going on in the department. And as Catherine just alluded to, Juan Perella and Jody Hayden-Perella are both involved in developing these really large computation models of very large macrocycles to understand the biological processes by, in some cases, predicting the structure, in some cases, refining structures based on NMR or cryo-EM data. Ed Lyman, who's associated with our department, is also in physics, uses similarly molecular dynamics and a combination of quantum mechanical and molecular dynamics tools to understand lipid bilayers and how lipid bilayers are influenced by their interactions with proteins. And so this is probably the kind of application of computational chemistry that most of you have been exposed to, solving the Schrodinger equation and doing molecular dynamics, using Newton to understand how atoms behave. On the other end, another application, another utilization of computers in chemistry comes from, let's thinking about the applications of Shannon James, right? And maximizing information retrieval from the chemical information we get from considering the structure, but also spectra and other chemical data that we might collect. And in that case, we want to be able to do things like classifying samples, classifying components of complex samples, being able to do things like track the components of complex reaction networks that have multiple species contributing to them. So all of these are examples of the kind of work that those chemical images come from Carl Busch's lab, and I'll be talking to you a little bit more about that. There's a new Raman microscope there that is producing these wonderful chemical images that we want to be able to extract more and more information from. And then we have a number of people doing spectral analyses that are producing complex data that we can mine more effectively. So this is the range of the kind of computational methods and tools that are being developed.
Thank you, Sharon. Okay, a focus on sustainability on our planet is also something that cross-cuts across many research groups within our department, both on the environmental side, environmental sustainability, and thinking about renewable energy conversion and energy sustainability as well. On the environmental side, we're tackling this as a department in a number of ways, right? Both in terms of trying to understand basic phenomena in our world that lead to different pollutants within our atmosphere, within waterways, et cetera. And so a really great example of that is the research that is done in Murray Johnson's lab. And so this picture is an example from an extended trip that Murray and members of his PhD students in his lab took a few years back up above the Arctic Circle in Finland. And so one of the things that the Johnson group is interested in understanding is the way in which aerosol particles within our atmosphere form, basically little sooty particles form from the combustion of fossil fuels. Okay, and one thing that's known is that important to that process is the combination of little small soot fragments with discrete organic molecules that are emitted from trees and other flora on the planet. Okay, and so what they basically did was to erect the scaffolding up in this forest in Finland and brought mass spec instruments to the top of this scaffolding to then directly analyze small molecule soot based clusters that form in real time to better understand what their molecular structure is, right? To understand the mechanism by which these larger aerosols ultimately form and then contribute to global air pollution. So very fundamental basic science to improve our understanding of how to achieve better environmental sustainability. We're not just interested in the air, right? Number of groups within the department are thinking about how do you either detect, build better sensors, or actively remediate small molecule impurities that might be in waterways. So I'll give you an example from my group. My group is building small molecule platforms that can bind and sense things like nitrites and nitrates, which are problematic oxidized nitrogen species, which are fouling groundwater runoff and are accumulating in aquifers based on heavy fertilizer usage with agriculture. And so we're figuring out new platforms to be able to detect levels of these species in these groundwater sources, and then be able to sustainably remove them, basically use things like renewable electricity or solar energy to convert those pollutants to value-added chemicals that we can use in the chemistry enterprise, right? We also can translate that approach to things like carbon dioxide reduction, thinking about global warming and climate change, and thinking about how do you actually draw CO2 out of the air and then either sequester it away or upconvert it into fuel precursors or other value-added compounds that can impact the chemical industry. So in many ways, environmental sustainability and energy sustainability go hand in hand. And to tell us a little bit more about other other research in our program that's germane to energy sustainability, I'll turn it over to Lars Gunlock.
Thank you very much. As Joel already mentioned, energy and environmental challenges are really closely connected. And you already saw on the previous slides that many projects, many groups are actually working on energy-related problems. So there are basically a couple of names for key faculty here, but I would say that almost half of our faculty is more or less connected to energy science. So I will just give you a very brief overview, really not pointing out names, what we are doing here in the chemistry department, working on these energy-related topics, which is, as you already saw, developing porous materials for gas storage that can help solving energy storage and distribution problems, synthesizing organic and inorganic materials for energy conversion and solar fuel production. We are investigating surfaces and interfaces to understand heterogeneous catalysis that is often key to solar fuel generation. And we're also studying charge and energy transfer processes to guide the design of new materials. And as I already mentioned, right, this is really interdisciplinary, and there are many collaborations actually in the department across campus, which also includes the Delaware Energy Institute that is here at UT. We can go to the next slide, please. So in addition to developing new materials for energy science, it's also extremely important to understand the detailed mechanistic processes of energy conversion in these materials. For example, if we want to develop new complex materials for solar energy conversion, there are a couple of requirements for these materials, and I'm going to cite now two of these requirements from a DOE report on grant challenges for basic energy science. One is to understand the remarkable properties of matters that emerge from complex correlations of atomic and electronic constituents. And another one would be to understand how to control material processes at the level of electrons. And the groups in the chemistry department here are approaching these challenges with a variety of tools. For example, by using electrochemistry and photoelectrochemistry to study redox potentials and energy level alignment in complex material, to measure macroscopic yields and reaction kinetics. On the top right of the slide, you basically see a couple of examples for electrochemistry and photoelectrochemical measurements. And we are also using heavily spectroscopic methods to study charge carrier dynamics in these solar relevant materials. And we will also hear a little bit more about spectroscopic capabilities in the department later on. I guess we can go to the next slide. So another very important ingredients, I would say, for solar energy conversion are interfaces and surfaces. And these are really essential not only for energy science, but also for a large range of applications in almost all areas of our everyday life. And numerous groups in the department are involved in surface science, studying, for example, how physical properties like chemical resistance or hydrophobicity of surfaces can be manipulated. This is this little black droplet on the surface here. How surfaces can be functionalized with organic or inorganic molecules to control their chemical reactivity, studying chemical reactions at catalytic or photocatalytic surfaces, and last but not least, also studying dynamics at biological interfaces like membranes here, top of the left on the slide. So these are all very active areas of research in our department, and this research endeavors heavily involve the spectroscopic and imaging capabilities that we have in the department and also in facilities across campus like the NanoFab and the Keck Center for Advanced Microscopy. So thank you.
Great, thank you, Lars. Okay, Jack Mugridge now is going to tell us a little bit about biochemistry, biophysics, and structural biology in our department.
Thanks, Joel. Hi, everyone. So I think, as you could tell from probably the past few slides that we've been through, right, we have many faculty in our department who are working on really important biological problems related and impacting human health. And so the faculty listed on this slide really use a wide range of different techniques from biochemistry, from biophysics and structural biology to answer some of these types of mechanistic biological questions. And so just again, a very, very few examples, just a small slice of what we're doing in the department here is, so for example, the figure 1 there is from Zhihao Zhuang's group. And this is work to develop different drug intervention strategies that target protein ubiquitination and deubiquitination pathways. So these are, ubiquitination are really important post-translational modifications that control protein function and protein fate. So for example, they're using PROTACs for targeted protein degradation and small molecule deubiquitinase enzyme, or DUB inhibitors that can be used to treat different neurological disorders and cancer. Number 2 is from Catherine Grimes' lab. And so this is some of their work to understand really the recognition of peptidoglycans in the bacterial cell wall by human proteins NLRP1 and NOD2, which play important roles in human health in terms of Crohn's disease and this skin condition vitiligo. Number 3 is from my own lab and our efforts to understand really the structural and molecular mechanisms that control these different readers, writers, and erasers of different classes of RNA modifications. So these are really important chemical modifications on RNA that control gene expression and impact human diseases like cancers like glioblastoma. And then 4 is a small figure from the Rozovsky Lab's investigation of really the biochemistry and the biophysics of selenoproteins. This is a class of enzymes that really protect our body from reactive chemicals that can lead to cancer and aging-related processes. And so really across all of these different research groups, we use a really wide range of different techniques like enzymology, cell-based assays, macromolecular NMR, protein crystallography, molecular dynamics, proteomics, and many, many more to answer these sort of important mechanistic questions in biology. And you'll hear soon about some of our facilities in a few minutes, but I'll just mention that we have really phenomenal new biochemical instrumentation in the department, including a brand new 600 megahertz NMR spectrometer for protein NMR, a new in-house diffractometer for protein crystallography. We're soon going to have a new Orbitrap mass spectrometer and proteomics facility director to support protein mass spec. And so it's a really exciting time to be doing biochemistry at UD.
Great, thank you, Jeff. All right, I'll shoot it back over to Eric, who will tell us a little bit about organic, organometallic, and inorganic synthesis.
Thanks, Joel. Yeah, so you all pretty much get the idea so far that, as synthetic chemists in the department, we're sort of tackling the classical problems facing chemists, right? How can we make new molecules to address problems facing humanity? So I'll tell you a little bit on the inorganic side. Mary will follow up on the organic side. These problems are classical problems, but our approaches to solving them, I think, are really anything but. So just an example here of a few of the pieces of work going on in the department along these lines. Thinking about small molecule activation, whether it's CO2 reduction, making fertilizer, thinking about making hydrogen from water. Here's an example of Joel Rosenthal making a palladium complex with this N-heterocyclic carbene ligands that can be used for CO2 activation. Just to the right of this, another similar and very challenging molecule, it's similar in that it's very challenging, is dinitrogen. So Klaus Leopold's low valent chromium complexes can be used to activate N2, which is a very important step along the way toward making ammonia, for example. Just above this, on the molecular side, I mentioned that Emil is making new nanoparticles, studying their formation, but he's really doing this from a molecular level, making new precursors that he can turn into nanoparticles with controlled size, shape, surface chemistry, and ultimately catalytic properties. And this feeds into a recent bit of my work shown just below this, is trying to use a molecular level understanding and just straightforward synthetic organic chemistry to design new molecules that can be used for host gas chemistry and catalysis. And Mary is happy to talk about some of the work that her and her other organic colleagues in our department are doing as well.
Yeah, I'm gonna get all organic for a second. If you're an organic chemist, sometimes just looking at molecules, right, they're just beautiful molecules, and thinking about how you control complexity and selectivity in making some of these is a really exciting pursuit. And so there's an amazing breadth of cutting edge research happening in terms of the synthesis of our complex organic molecules. So this slide shows you some of those examples and some of the different applications or reasons that we want to make these. So if you look in the upper left, this is a molecule from BJ Chain's lab, where their lab really focuses on synthesis of complex natural products. This is Ciguadiol A. And in particular here, the formation of the 7-membered ring in the middle of the molecule presented real opportunities for creativity and innovation in the Chain Lab synthesis of this. If you go just to the right of that, you see a different example. So these two examples in patient A and octreotensumine are both molecules that Don Watson's group is interested in making through novel chemistry that they created, the AZA-HEC reaction, which is one of the first examples of being able to use heteroatom nucleophiles in these palladium catalyzed cross couplings. If you go to the structure on the bottom left, this highlights actually a totally, well, to my eye, different sort of natural product. And so Professor Catherine Grimes' lab focuses, as you saw before, on these sorts of glycopeptide molecules that are involved in bacterial cell wall synthesis and also inducing the innate immune response in humans. And so her lab has developed some really beautiful syntheses of these types of molecules, which are just as complex and in some ways harder to handle because of their polarity than some of the natural products that are, you know, kind of classic targets within organic synthesis. If you look at the bottom, the square, you may recognize some of these types of molecules. So from earlier slides, so you've seen how Professor Rosenthal's research has focused on using solar energy to combat our energy problem. And as part of that, his group actually does quite a bit of organic synthesis, and their target are these designer tetraperols. And again here, controlling efficiency in synthesis and also selectivity in synthesis has been something that they've really had to bring innovation into their synthetic schemes to accomplish these. If you look at the bottom right, this is actually an example from my group, and you know, maybe the most simple example on here, but it shows you just another way that we think about making molecules. So my lab thinks about late stage functionalization. So you may recognize the red part of this structure actually comes from Tamiflu, and we didn't make it, we bought it. So my lab makes complex molecules by buying them and then transforming them into different molecules that may have different biological activity. And so that's an exciting thing that we've been able to do. Just like every other slide you've seen through this deck, these are just a few examples of how chemists and biochemists in our department are leveraging synthesis to solve research challenges. And so I really hope you'll reach out and learn more about all the people on this slide.
All right, thank you, Mary. And the last topical area that we'll cover is spectroscopy and imaging, which Sharon, you'll tell us about.
Oh, thanks, Joel. My charge is just to give you an overview, and we have a really wide range of spectroscopy being carried out here at UD. And so we have work being carried out across the electromagnetic radiation spectrum from very long wavelength NMR that you're gonna hear about, the world-class NMR facility, and which is, and that's not all the NMR that's going on. A number of people have dedicated NMRs for their research, but from long wavelength NMR to high energy radiation to characterize materials, to ultra fast optical spectroscopy, which I'll say a little bit more that goes on in the department. And so the pictures here are just give you a snapshot of some different work that's being done. Mostly that we picked the pictures because we thought they look good, but let me just highlight a few of the examples. So for example, Sharon Rosofsky and Cecil Dabowski use unusual NMR. So you probably done carbon and hydrogen NMR at your schools. But Sharon Rosofsky's lab uses selenium-77 because it's a good surrogate for sulfur, and it allows her to understand the processes that proteins that are typically sulfur containing. She's transformed them so that they're selenium containing, and then she can use the selenium NMR to monitor their processes and understand the biological function of those proteins more completely using that technology. And similarly, Cecil Dabowski uses heavy metal NMRs, many types of lead and many other nuclei to characterize materials. And so those are, in addition to the sort of normal NMR that you've been exposed to, we have this exotic NMR going on here. We also have interesting surface spectroscopy, and I want to just highlight the work from Andrew Tepliakov's group. They're developing really precise, atomistically precise functionalization reactions, and they use vibrational and electron spectroscopies to monitor the progress of those reactions and to determine the success of functionalizing the surfaces using their new reactions. And then Lars Gunlock's group is the ultrafast optical spectroscopy group that I was referring to, and you can see that picture of a nanoparticle, that rod-like nanoparticle that they've characterized. And they can study both heterogeneous electron transfer in these interesting materials and other kinds of charge carrier dynamics in nanomaterials on these really short length scales and on very fast time scales, characterizing processes that are complete in less than a picosecond. I mentioned to you earlier the new Raman microscope that is now set up in Carl Buch's lab, and that's being used to investigate a number of novel materials and unique materials. One is not novel, but still really interesting. They're beginning to study Mars rocks and be able to characterize the composition there. And you can see that we have many other kinds of materials and spectroscopies that are being used. But that's just an overview of the work that's being done in the department.
Okay, thank you, Sharon. All right, so we're going to switch gears slightly at this point. We're getting close to the end of the slide deck, and now we'll just relatively quickly go through the resources, the instrumentation, and the core facilities that enable us to attack the areas of science and many others that we just showed you in the last slide. So, and I'll also mention that on the Perspective website that we went through at the opening today, there are videos and specific overviews of many of these core facilities, of many of these major instrumentation labs as well that I encourage you to check out. So the first we'll talk about is our NMR core facility. And our NMR facility, really, taking a step back, I would say our instrumentation and core facilities in general are outstanding. And I don't say that lightly, So before I came to UD, I was at the Massachusetts Institute of Technology for both my PhD and postdoc. And I was at the MIT, so I figured the instrumentation we had there was pretty good. And it was fine. We did all the work we needed to do. But then I came to UD, and it's really not aligned. The capabilities and the resources we have here are second to none. When we can have these prospective open houses in person, the facilities tours is generally the first thing we do with the students that are visiting our campus over the first 45 minutes or so. In general, the students come back and they all come back like this, because they've never seen the NMR, the mass spec, the crystallography, the materials characterization. So they've never seen all of it, so much of it all in one place. And NMR is certainly in that model. If we have a crown jewel as far as all our core facilities go, NMR might be it. We have a full 7 NMR spectrometers within our NMR core facility, ranging all the way from 400 megahertz up to 850 megahertz. Some of the instruments have specialty probes that allow us to, as Sharon mentioned, that allow us to look at nuclei that most researchers simply do not have access to at their institutions. And that's both on the biochemical side, on the material side, but also on the organic and inorganic discrete molecular side as well. Our NMR facility has 2 dedicated PhD level staff scientists and a graduate student who serves as a laboratory assistant for that facility to help keep it running. And more importantly, right, our 2 PhD level staff scientists, they are world-renowned experts in the application of NMR spectroscopy to answer all kinds of questions in chemistry and biochemistry. So, not only do we have the physical tools, the instruments themselves, we have the intellectual resources of Steve Bai and Caitlin Quinn to help us ask questions using the instruments that we have that many researchers at most places simply can't do. NMR is simply outstanding. X-ray crystallography and X-ray diffraction. So our X-ray core provides high resolution structures of organic and inorganic small molecules and even large biomolecules as well. We have capabilities to do both single crystal X-ray diffraction and powder diffraction for characterization of different materials. We have 2 single crystal X-ray diffractometers at present, including one brand new Bruker D8 diffractometer, which allows us to analyze not just small molecules, but also larger macromolecules and even some proteins and biomolecules as well. This new Bruker D8 diffractometer has a super intense X-ray source and incredibly sensitive detector, which enables us to actually do the analysis of some of those larger systems, the macromolecular systems. But on the other end of things, right, it also allows researchers that grow crystals maybe of smaller, more discrete organic molecules or traditional inorganic complexes to be able to analyze crystals that 5 years ago, a decade ago, we never would have been able to mount on an instrument and get a reasonable data set off, because our older instruments simply didn't have the power, didn't have the sensitivity to get a data set with enough fidelity. Our instruments have both moly and copper sources, and again, just like NMR, our X-ray facility is staffed by a full-time PhD staff member, Dr. Glenn Yap. He is a world-renowned expert in X-ray crystallography. He is one of the editors of Acta Crystallographa. He is an amazing resource to us as well in terms of collecting our X-ray data and, most importantly, solving X-ray structures that often prove very difficult to solve. Our mass spectrometry core facility is an area that's really grown remarkably over the last 5 years to decade. It has 10 mass spec instruments. It's also staffed again by 2 dedicated PhD staff members. One is Dr. Papani, whose picture you see here at the top, and Dr. Jiha Yang as well. And so in addition to the excellent instrumentation, which includes walk-on and high-resolution GCMS, LCMS, MSMS, and PrEP LCMS options, we get to take advantage, and our science is sort of brought up to the next level by the expertise of these 2 dedicated staff members. We never are complacent. We are never content to just have a facility and accept the status quo. We are constantly trying to write grants, get resources to bring the next generation of bigger, better, more efficient instrument to all our facilities. And as Jeff Megridge alluded to when he was speaking to you a few minutes ago, mass spec right now probably leads the way in this regard. A brand new Orbitrap is coming to campus. A brand new proteomics core is being built out with new instrumentation and another new PhD level staff member to lead that. So mass spec is an incredible tool that touches all areas of science and is something we can rely on in our department. The surface analysis core facility Again, very, very important for both soft and hard materials related research and much biochem and more molecular research as well. Surface analysis tools within this core facility within our Department of Chemistry and Biochemistry include a relatively new X-ray photoelectron spectroscopy instrument, a time-of-flight secondary ion MS instrument, of which we have currently written a grant and are trying to bring an even newer generation one of those to our campus. And as Sharon Neal alluded to, a tandem AF Raman microscope that is really powerful and facilitating lots of important work at the materials interface. Our SAF, our Surface Analysis Facility, is directed by Professor Tom Beebe and also has a graduate laboratory assistant. Many, if I forget to mention it, many of our core facilities within the department also have a graduate laboratory assistant that helps run it. Another thing which I want to make sure I mention, I'm not sure I did before, is that the way our facilities, our core facilities are organized are not such that these instruments sit behind, a glass wall where you look at them and, you submit a sample and then you walk away and you get data. There are some instruments and some instances where that is the mode of operation, but, there are many instances where it's important for the student or the researcher who's actively in the lab trying to drive their science forward to actually have the capability to collect data, whether it be NMR, whether it be mass spec, whether it be XPS, on their own, right? So that they can design the experiment to meet their specific needs, and then get the data right away and be able to interpret it and say, oh, well, this isn't quite right. Maybe I need to tweak my experiment in this way, and recollect. And so, one of the hallmarks of all our facility is that the vast majority of our instrumentation is available for students to be trained and use it day to day, right? It doesn't sit behind a locked door. It's not just there to be seen, submit a sample and get your data, right? Students have full access to use this very, very high level instrumentation, not only to better drive their research forward, but to gain real technical competence in the use of those instruments, which then serves them as they earn their PhD and move on to other things once they graduate. Okay, some additional core facilities which are important to mention are the WM Keck Center for Advanced Microscopy and Microanalysis, the Advanced Material Characterization Lab, and the Bioimaging Center at the Delaware Biotechnology Institute. These all three of these instrument centers reside in buildings that are right by our central chemistry and biochemistry labs. The Keck Center for Advanced Microscopy and Microanalysis and the AMCL are basically, they are in an integrated science and engineering lab that is directly adjacent to Drake Lab, right across the street there. The Keck facility is a top-notch facility for advanced microscopy. It provides us access to multiple transmission electron, multiple scanning electron, and scanning probe microscopes, along with other pieces of complementary equipment for materials characterization. And just like the facilities that I mentioned in our home chemistry department, these are all high-level instruments that students get trained to use, right, and collect data on themselves, right? They do not sit behind a locked door. The Advanced Material Characterization Lab in the same building, basically right across the hall from the Keck facility. This lab features a full array of equipment for X-ray analysis, thermal analysis, certain mass spec experiments that complement what we can do within our own department, Raman and IR analysis, as well as resources to probe materials porosity, the zeta potential of different materials, and the particle size of materials. And then down in the Bioimaging Center at the Delaware Biotechnology Institute, this is right near our Biopharmaceutical Innovation Center. It's a resource that any member in our department can have full access to. This includes equipment ranging from confocal to super resolution electron and AFM microscopes, And so this center, it really enables researchers to probe samples all the way from the millimeter scales, right? You can see this basically this micrograph of a ant shown here on the left, all the way down to the nanometer scales. Okay. One final facility that I will tell you about is the high throughput experimentation facility. This is another facility that resides within chemistry and biochemistry. It's relatively new. It was established just a few years ago, and it's a facility that's designed and intended to streamline the discovery of new reactions and new ways to make new compounds. Okay, and it really can facilitate any project that requires synthesis. Okay, this center again has a full-time PhD staff member in Dr. Austin Kelly, who serves as the director facility and will assist any student that needs access to the HTE and its tools. The facility features this mammoth 25-foot glove box here that has basically a library of different pre-plated ligands and metal salts, right? And so the way it works is, if you're trying to think about maybe a new way to stitch together an olefin and an aryl triflate, right? Basically trying to do a take on a Heck reaction, but maybe using two substrates that are proving really difficult to get to couple in that way, right? You can add those coupling partners to each well in a 96 well plate, and then systematically change the types of catalyst, the loading of catalyst, the basicity in each one of these plates, in each one of these wells, and then basically be able to run all those reactions in parallel at the same time, and then take advantage of instrumentation in this resources that's basically involves robotics and automated analysis of how the reaction is progressing in each one of these wells to see how changing the catalyst, changing the loading, changing the basicity, changing how much ligand impacts how efficient that reaction works. So it's a way, right, instead of linearly running 96 reactions to figure out, as I change these parameters, does the chemistry work or not, to do it all at once and use tools of robotics and automation to go through that data much more quickly to basically find the needle in the haystack, if you will. And I was wrong, there is one more, Lotus Separations. Lotus is another really important facility that we have on our campus, and one that I think really distinguishes UD in our department. So Lotus is a collaboration between UD and this company, Lotus Separations, which brings preparative supercritical chromatography to our department. So, and there are basically 3 universities that I'm aware of, only one other of which is on the East Coast, and that's Princeton, that has this capability. Okay, and so the way this arrangement works is that researchers from our department can have any sample at all for the most part that needs to be purified and separated. Maybe you run a new kind of reaction, you know, and based on a crude NMR, it looks like the reaction works, but it's really messy, and you know, a lot of the products just it's a huge pain to separate them using conventional chromatography. They just won't separate using those conventional techniques. Well, we can essentially take them down the hall to Lotus, drop them off, and they apply their incredible expertise and their world-class resources in supercritical chromatography to essentially, 1, figure out a method to purify what we want out of that mixture, and then 2, do it for us. So you can run down the hall with a vial of just the grossest looking tar that comes out of a reaction you try that might have the product you want, drop it off, and within, you know, a few days to a week, this happens routinely. They will return a vial to you, right, that has the brightest, cleanest, microcrystalline pure white solid that they've been able to isolate using their methods. So this is a tool that enables us to do purifications and gain access to compounds that we would not otherwise be able to do, and that most researchers across the country don't have. And then finally, departmental shops. So in addition to the core instrumental facilities that we talked about, we're very lucky to have additional dedicated staff and dedicated shops to assist us in our teaching and research missions. And so these include computing and IT, and that's everything from, my computer crashed and, or, my latest Mac update, something went awry and now I can't open Word, to figuring out how to set up a new DFT cluster in a specific research group. So we have two IT personnel, Pat McMahon, who is just an incredible resource for us. He can basically do it all. I've never had a problem that Pat couldn't fix, quite frankly. Is the main driver there. We're very lucky in that we have a dedicated glass shop. So, if you have a flask that breaks or you crack your strength line, you say, oh no, what am I going to do? We're lucky enough that we don't actually have to send our glassware out, right? We don't have to ship a piece of glassware and then wait a month and a half for the fixed item to come back. We can run it down the hall, drop it off, and usually, sometimes, often in as soon as a day or a couple of days, that glassware will be prepared. Having a glass shop and a dedicated glass blower on site also gives us the flexibility to design new apparatuses, right? To design a piece of glassware to do a specific kind of experiment that we might not be able to do otherwise. We can run down the hall, talk to the glass blower, sketch this all out in paper, and then buy the glassware, the blanks, and they will basically put it all together for us. Similarly, we have a full-blown machine shop with a full-time machinist to build other pieces of equipment or other apparatuses that we might need for our research and teaching, as well as a dedicated electronic shop, which is great not just to repair instrumentation when it might break, because it is increasingly more and more and more electronics based, even something simple like a gravity oven. But on the other end, it also allows us to think about maybe we need to build an instrument or build a tandem instrument to do a specific experiment that we might not otherwise have access to. The electronic shop helps us in that regard as well. Our chemistry library and the subscription service is really top notch. For the most part, over my time at UD, there has very, very seldom been an article in a journal that we have not had just inherent access to through our library service. We really have an awesome subscription service. We also have subscriptions to all the major chemistry and scientific databases, Reaxys and SciFinder being two of them. And we have a full-time chemistry library staff member who's basically partially responsible for serving the needs of our departments. We have a direct liaison for any questions that we have. All right, so I think that's a pretty good summary of the resources. So we're going to switch gears now, and I will again toss it to Catherine Grimes, who will tell us about the Chemical Biology Interface Program.
Thanks, Joel. So I hope you get a flavor for all of the amazing resources and facilities that we have on our campus. And I just want to tell you about a program that we have on our campus that might be interest to you. Is at the Interface of Chemistry and Biology. It's a prestigious NIH-funded program that is embedded within the CBC community. And I'm lucky enough to lead this highly dynamic program, which unites programs from chemistry, biochemistry, biology, and engineering under a common training platform. So we have a dynamic seminar program, we have fellowship opportunities to support your research throughout your time at the University of Delaware, and we offer lab rotations for our first year fellows. I'll highlight more about this program on Wednesday, as Joel mentioned at the beginning of this overview, around 1 o'clock, and you're welcome to join us for that seminar beforehand. And so maybe Joel, you can go to the next slide. So regardless if you're not interested at the Interface of Chemistry and Biology, just as we've been highlighting the prestige of our university, this puts us on a map with other universities. We're one of a few programs that have this chemistry biology interface training grant in the country. We were actually one of the first to have this program. So we're really innovating the way that people think about cross-disciplinary research. So just to put this in perspective for you. And so Joel, you can go to the next slide. But this is really largely driven, the ability for us to land on that map is driven by other centers that are focused on bringing major NIH funding for our research to campus. And so two of these centers are the Centers of Biomedical Research Excellence, or COBREs. And we're lucky enough to have two housed within our department, one led by Tatiana Polanova, which focused on the design of molecular design of advanced biomaterials. And it really brought and innovated many of the resources that Joel just talked about, such as NMR and mass spec. It was those research dollars that allowed us to innovate and update and upkeep our instruments that we have available to you for your research on campus. And then we have a newer COBRE led by Professor Joe Fox, which is for the design of the discovery of chemical probes and therapeutic leads. So really honing in on thinking about therapeutics to treat human diseases, making these small molecular probes. And so this was just renewed, and it's really dynamic program. And it again impacts the way that we think about and run our research programs. And so these are two of the centers. And I believe that Mary's going to talk to you about some of the other multidisciplinary centers located across campus.
Thanks, Catherine. So in addition to the CVI program and those two COBRE centers, there are a tremendous number of other multidisciplinary centers here at UD. And I just want to point out, I won't go through them one by one, but these centers target a range of global and societal challenges, ranging from the energy crisis to human health to global warming to the plastics waste problem. So they're coming together with this multidisciplinary approach to really be able to solve real challenges for our world. And very excitingly, chemistry and biochemistry faculty and students are involved in every single one of these efforts. And this gives exceptional training opportunities to our students. And I'm really excited about that. If we go to the next slide, I'm just going to briefly touch on another initiative that I'm excited about within our department. And this is an increased focus on creating an inclusive, equitable, and diverse community of scholars within chemistry and biochemistry. So, the summer of 2020, I think, really brought a lot of the inequities in our society to the forefront of a lot of our minds. And at that time, our department established a new committee, which was the Committee for the Allies for a Culture of Inclusive Diversity, or ACID. And so the ACID committee is leading the effort within the department to bring attention to creating an environment in which all members of our department can flourish and thrive. We've had a number of initiatives over the last year, including seminars and workshops, but also just community building events like a Halloween movie night. And we're really excited to get to talk with you about the community and culture and climate within our department. And also just get to know you tomorrow at the coffee chats in the afternoon.
All right, and so the last thing I want to leave you with was something that I alluded to at the very bottom of the first slide, which was providing opportunities for our students to essentially be able to apply what they are learning in a real world setting. And so both students and faculty within our department benefit immensely through interactions with industrial colleagues in the Mid-Atlantic region, of which there are an immense number, right? I mean, the region in which we sit, it's a hotbed, it's a hub for pharmaceutical science, for chemical synthesis, for biotechnology, and for many, many other chemistry-related industries as well. And so a lot of our students get to take advantage of these connections to engage in internships and cooperative experiences with a mix of both industrial and national lab partners as they complete their dissertations, okay? And so what these experiences do is they provide a student with, I think, a much greater understanding of how the skills they're building translate to the workplace as they prepare for the next stages of the career, right? It also is a great way for those students to be able to, build up their professional network while they're still a grad student, right? Either within that industrial setting or at the national lab. And so often the time spent during these internships or cooperatives, they're setting the stage. They're building the network that that the student will take advantage when they're ready, once they receive that PhD, to go on to that first career opportunity. And shown here are logos for just a few, just a small fraction of the companies, the national labs, the other institutions where our students have gone and done internships or had those cooperative experiences in the past. And I'll also note that many of these companies, many of these different places also will routinely visit our department, okay, to meet and interview our most senior graduate students as they get ready to start their independent career. So it really benefits our program and the students that make up our program in a number of very, very meaningful ways, okay? The final thing I want to leave you with is more or less what I started with, right? Our philosophy and our priorities. Our goal at the end of the day is the success of our students, okay? We measure our success as a program, right, by stressing the success of all of you when you come here, right? And one doesn't happen without the other, okay? We cannot be successful if our students are not successful. So that's what we work toward on a day, week, month, year basis, right? And so just to give you an idea, because rightfully so, right? This is something that prospective grad students ask about, they think about, you know, if I come to the University of Delaware and I get my PhD, work toward my PhD and earn my PhD with you, you know, what do I do with that? Where will I go? Well, if you look at employment numbers for the students that emerged from our program over the last handful of years, okay, this is what the numbers look like. This is students that emerge with their PhD within 3 months, right? 2006, 100 of them had full-time jobs. Same in 2017. That number in 2018, I'm pretty sure is simply because one person didn't reply to repeated queries. And 2019, and we're building the data for 2020 now. Here's sort of a snapshot of where our students go, what sectors they go. This is the data from 2019. It moves around a little bit every year, but this is a pretty good snapshot. Roughly a third will go on to an academic setting. That means either directly to a teaching job or maybe a postdoc at a university if they want to stay on an academic track. Roughly 50 to 60% in any given year will go directly and apply their PhD to an industrial job, and 15 to 20% in any given year will go to a national lab type setting. Here are some of the top employers integrated over these last 4 years from which I'm showing you data where our students go. So you'll see that there are a lot of very notable names on that list, right? Very, very high ranking universities as well. There's an awful lot of star power and name recognition on this list. So the bottom line is, with a PhD from chemistry and biochemistry at UD, a student can go wherever they want to go. And the philosophy of our department and the way we structure the graduate program at our department is to think about day in and day out and work day in, day out to help our students get ultimately where they want to go. Okay, so I'll end there. Open it up for any questions if there are any. I'll note that, you know, if you missed anything or you need to go back, a video of the presentation that we just went through will be posted on the Perspective Information website. There's those additional resources on the bottom. And if you have questions for me or any of the other faculty that you heard from today, we would be delighted to hear them now and give you whatever information would be helpful.
I've got a question.
Sure.
So for those graduate lab